So the first thing I want to go through is getting everyone on the same page. How many of you have, before working at Twist, have had experience in the NGS field? Yeah. So you'll learn very quickly about kind of the beginnings of NGS and about kind of you'll get like really good deep dives in NGS. But for today, I really just want to get like something very high level. What is an Illumina sequencer? What is a PacBio sequencer? What's the difference between them? Um, so this is just going to be a very high level kind of basic NGS 101, just to get everyone on the same level. So when we talk about some of our products, we're all on the same page and we all know what we're talking about. So um, by the end of this lesson, we should be able to understand the basic principles of NGS, which a lot of you probably already do. Explain the difference between NGS and other traditional sequencing methods. We have had a lot of people come to us, what's the difference between Sanger versus NGS, or the different technologies out there. Identify key components of the workflow. This is actually quite important for understanding what you're selling and what you're supporting. So I'll go into a little bit more detail there. And then we're going to discuss some real world applications and research in healthcare, because at the end of the day, it's all about helping people. And what's really cool about NGS and DNA sequencing in general is you're taking something biological and making it digital. So that technology we take for granted a little bit, making anything digital, from something like going from VHS, right, to DVD. I mean, we take a lot of that stuff for granted, but there's a lot of crazy technology that's going on, and this is a crazy world we're living in to be able to do this. So, all right. Has anyone seen The Central Dogma of Life? We'll go back to biology, right? So I don't really have to go over to this much too much. So this is generally just saying DNA gets transcribed to RNA, RNA gets translated to protein. DNA is the code for making you, right? RNA is what gets kind of transitioned into the protein. The protein is the function of what makes you. So code, application, function. So with RNA, you can use something called reverse transcription. So this goes back and forth all the time, right? In the body, you generally don't get RNA to RNA to DNA, because we don't have RTs in our body, reverse transcription enzymes in our body. But there is, when you start looking at like library preparation for RNA, you need to convert it to DNA. So this is also kind of an important step here. Also, as we all know, cells replicate, so DNA also has its replication. So this is the central dogma of life. This is what makes everybody and every organism on this planet work. All right, the field of genomics, right? So when I first did this, I feel like there's different levels of kind of experience here. So I'm sorry if I'm not trying to talk down. I'm not trying to kind of, but I'm trying to make sure that we're all on the same page. So genomics is the complete study. It's a study of complete sets of genes in an organism, and understanding its function and mapping across genomes. When you start looking at DNA and RNA, actually, then RNA is the next one. There is a slight difference in the compound of DNA. There's a slight difference in what makes DNA in terms of the nucleotides that are available, right? So DNA is made up of adenines, thymines, guanines, and cytosines, right? So this is, again, going way back to potentially high school. But the genomics is the study of DNA and how it's working in the body and outside of the body. So there are different elements of the genome, right? There's a lot of different components in the genome. When we had the Human Genome Project, we were like, great, we cured cancer. Yikes. No. So there's a lot more to the genome than just the DNA base pairs. There's base pairs. There are gene bodies. There are intronic and exonic regions. Those are coding and non-coding regions in the genes. There are histones that DNA wraps around very tightly and make DNA accessible, which means that it can be used to make RNA. There are also, that's not on here, there are modifications to the DNA that are beyond the histones. So these are called the epigenome, which I'll talk about in a second. Everything is tightly bound in a chromosome, and those chromosomes are what are packaged in a cell. So we have something like 3 billion base pairs of DNA in our system, and every single cell in your body has the same exact DNA, which is crazy, right? If you think about it, we have different body parts, we have different organs, we have eyes, we have hair, we have nails, right? We have all these different things in our body, but every single cell has the same exact genomic makeup. And obviously, DNA sequencing is determining the exact order of the bases of a strand of DNA. And when you do the DNA sequencing, you're looking at single base resolution. So you can actually physically read the adenines, guanines, thymines, and cytosines. Some implications of DNA sequencing in the medical science. So precision medicine is really big, obviously. Making treatments and drugs and outcomes, diagnoses, basically making these medicines and treatments for people that are tailored to their genome or tailored to their genes. That's a big science right now. Think MRD, think CRISPR, which is a little bit on the gene therapy side. Diagnosis and risk prevention. So if you're looking at cancer diagnostics, you're looking at prenatal screening. These are different applications that have been applied to NGS that actually Twist has been involved with as well. So all these things Twist has actually been involved with. Genetic disorders, there's a difference between something looking at what's called somatic versus germline. So if you're looking at genetic disorders from your parents that are inherited from your parents, you're looking at germline conditions versus what's called somatic or tumor samples or identification. So there's just a whole bunch of things you can do in the DNA world, right? Infectious disease and surveillance. That was big during COVID, right? We're trying to make sure, and that's still big. You still have a ton of people looking at wastewater, right? That's a huge thing. One of your biggest customers, the Lipkin lab, they are doing wastewater identification. They're doing like viral surveillance. So making sure that there's a healthy level of DNA in wastewater. If it spikes somewhere, then that's maybe not a good thing, right? And you can do that through detection with NGS. And then obviously gene therapy, also a big up here. Agriculture, I don't really have as much here because my experience is mostly in Boston, which has a lot of gene therapy. Gene editing therapies and identification of on and off targets. So I'll go into that a little bit later as well. So transcriptomics, RNA. So transcriptomics is looking at the transcriptome. So as I mentioned back in that the dogma of life, DNA gets transcribed into RNA, right? You're looking at, usually you can directly correlate the transcription or the generation of RNA to direct kind of phenotypic. So that you're looking at the genotype versus the phenotype. Looking at the RNA, you can virtually make a direct correlation with as much, if there's a lot of RNA being transcribed for gene X, there's probably a lot of protein for gene X or dysfunction of protein for gene X causing some condition, cancer, whatever. RNA is slightly different, if you remember, than DNA. There are adenine, guanine, cytosines. There are no thymines. They're uracils, right? So there's a slightly different in terms of the nucleic acid makeup. Does anyone else know what the other main difference with RNA is than DNA? Got it. Single-stranded RNA is DNA is a double-stranded double helix, whereas RNA is a single strand that goes out into the ribosome to make RNA, make protein. And of course, RNA sequencing is you're not just looking at the sequence. So there's a lot of different applications for RNA sequencing. You're not just looking at the nucleotide resolution with RNA. You are looking at kind of the abundance of transcripts. So transcripts, again, are those transcribed portions of DNA into RNA. And if you quantify those transcripts, those RNA molecules, you can make a direct correlation to a phenotype, generally. Kind of went through that pretty quick. Any questions on DNA, genomics, RNA, transcriptomics? So are we all kind of good there? We'll go into a little bit more about like the products that support these studies later. There's some implications. Again, there's always disease diagnostic and risk prevention, right? If there's looking at cancer, if there are, so there are things called oncogenes, which I'm sure everyone's aware of. These oncogenes are good at, so what they're typically for are expression of oncogenes causes the repression of certain cancers. So if you have any kind of dysregulation or changes in that RNA or even that DNA, then you can have, I don't know, muscle cells, lung cells, blood cells. You can have all these different tumor types that are just growing in your body, right? So because all your genes in your body have the same exact genomic makeup, you can have them go haywire in a lung, in a liver, in a kidney, in your blood, in your brain. So all these different genomic makeups that are accidentally being activated, that's a tumor. Cancer is just your body's own mechanism going haywire, being dysregulated. RNA sequencing is very important in kind of identification of what's being dysregulated, right? We can also look at other disorders like genomic, like neurological disorders and kind of inherited diseases as well. So, have any questions with that?
So if somebody has breast cancer, but they find breast cancer cells in their back, how does that tie into this?
And they find it in their back, is that what you're saying? So that's called metastases.
So I know it's just like the breast cancer has spread, but does that mean like how does that work with this?
Yeah. So okay, that's actually a really good question because that's starting to get into somatic detection of tumors, right? So say you have, a tumor growing in the breast, then those cells, so tumor cells are very fragile. So similar to your when you broke things up and kind of homogenized your cells can break off of a tumor and then travel somewhere else in your body. That's called a metastasis. That means your cancer cell in your lung has broken off and traveled somewhere else in your body and latched onto anywhere and started growing. So at that point, that's like stage 2, I believe. Stage 2 cancer is when it starts metastasizing. So they're breaking and moving around. And so when you start looking at circulating tumor DNA, those are also cells breaking open and releasing their DNA. Or cell-free DNA is also, you see a lot of people looking at cell-free DNA. That's just like broken open cells releasing DNA into your bloodstream. But also cells themselves can break off and float around, land and implant somewhere else and start growing, and like your leg or your bones, which is even worse. Lymph nodes are a really great pathway for cells to travel in. You get cells in your lymphs, then I think that's actually stage 2 is when it starts getting into your lymph. Because that can travel anywhere.
They'll be like, well, we don't want to cut because there could be a greater risk of them causing cells. Is that generally based more on the cancer or more on the region that they need to get into that it just makes it more dangerous?
A little bit of both. So what happens with tumor cells, they're really great at something called angiogenesis. So cells need blood. They release cytokines, they release other hormonal markers and other indications that cause arteries and veins to grow. So they start creeping out toward the cells or toward the mass or the tumor cells, and they start wrapping around, and they get pretty big. So if you cut some things out, you could cause internal bleeding, and the patient can die. So that's why a lot of people use radiation first to shrink it. Hopefully that angiogenesis starts getting kind of broken away through chemical or radioactive means, and then they can extract it. But there's also you could also cutting these fragile masses away could release some cells. And that's when you start looking at MRD, right? You're looking at minimal residual disease. You could still have some of this DNA or cells floating around your body that after the extraction, it could still be there.
I know there's a huge risk in that for women.
Yeah.
And certain types of cancer because they don't realize it's there
and
it's not your regular trained in that. They go in.
Yeah. Yeah. Breast cancer is very susceptible to that because there's a lot of lymph nodes in your breast. So that could get into your lymphatic system and travel anywhere in your body. Yeah, great discussion. That's great. All right, epigenomics. Now we're getting a little bit more complicated. So again, when they thought that when they sequenced the human genome, they're like, great, we can cure cancer. We know all the bases of the genome. We're golden. Let's do it. Little did we know at the time that there are DNA modifications that are causing the actual regulation of genes. So we thought, oh, gene X does phenotype Y. There's a bunch of other genes that do a lot of things. There's redundant pathways. There's things that are activated, deactivated. So the field of epigenetics or epigenomics is looking at modifications in kind of the makeup of DNA beyond the genome, epi beyond the genome. So we're looking at things called histone modifications. So remember those things that the DNA wraps around very tightly? Those could be either wrapped together or they can be open based on the epigenetic makeup, based on these molecules that are attached to those physical histones. Those are just giant proteins, right? That the DNA is wrapped around. There are modifications to that. There are certain molecules that are added to those histones. Where we shine at Twist is we can do detection of modifications directly to DNA, right? So we look at what's called CpG methylation. And what that means is there are methyl groups, just like methanol, a CH3 group that is physically attached to a cytosine that can cause issues with regulation. So if they're in general, when there are more methylated DNA fragments or methylated base pairs together, it causes chromatin to shrink up and kind of bind to each other because it's very sticky. Once there's less of that methylation, these tend to open up and become more accessible to transcription. So once you have DNA open and available for what's called a polymerase, an RNA polymerase to physically bind. Once that binds to that and it goes up the strand, along the strand, that's called transcription. That creates RNA. When you have the DNA open and accessible, you have it more able to bind to that and start that transcription. Whereas when everything is closed and tight, it can't do that, so you don't have transcription. So there are many different techniques that are out there that you'll hear. You'll hear customers, especially in the academic field, you hear a lot of cut and tag, cut and run. That's looking at histone modifications. We don't really do too much of that, but we can do the downstream like library preparation for that. So if you hear someone say cut and tag or cut and run, or even attack seek or chip seek, you might hear a lot of chip seek, right? Chromatin immunoprecipitation. Don't just run away from that. Think that there is a downstream element that can be used for that. So, but really where we live is this bisulfite sequencing, or in the case of what we sell, an enzymatic methylation sequencing. So again, yeah, there's a lot of different techniques out there. Make sure, please always ask your FAS or the technical team if we can support anything in this methylation space, because there's a lot of different methylation detection systems and kind of epigenetic systems out there. So just, make sure to ask. We can support anything. If someone does any kind of bisulfite sequencing, we can make panels for that. If there are any kind of, if someone is using the NEB EMC kit, again, we can make panels for that. We supply it. If someone is doing ChIP-seq, even at a single cell, we can do library prep for that. Well, not the single cell library prep, that's something else, but we can do library prep after what's called the immunoprecipitation. That's getting kind of technical, but we can do a library preparation for that after that precipitation. The mechanical fragmentation kit. Because
why it would be mechanical versus enzymatic later.
I won't, it's not on the syllabus, but like I can say. So the reason being is you already have to do a mechanical shearing upstream of the precipitation. So how ChIP-seq works, it uses an antibody. So antibodies are shaped like a Y. it will physically bind to one of the chromatin modifications and yank that piece out. So it's like doing an enrichment for that. But you have to chop up the DNA first to be able to do that. So that's the first step, is to do a mechanical shearing. So then once you do that mechanical shearing, they do the precipitation, they capture that DNA, then you can go into the mechanical fragmentation kit.
I've seen studies where it's like, I think there was one, like the potato famine or something. And
Okay. Yeah.
Someone who was pregnant with a child, that then transferred to the child, and then they could see it in other down the lineage or whatever. So once it's affected, would that always transfer down, or it could or couldn't?
It's funny to bring that. That was one of the first projects I worked on at the Broad, was part of the potato famine, and looking at the epigenetic profiling of the grandparents, parents, and then the children, and it actually skipped generation. So the epigenetic profile. So the Dutch famine was a bit of a tragedy, obviously, in Europe during World War II, I believe, or World War I, I forget, where they were on strict rations in Netherlands, very strict rations, and people were starving, people were emaciating, people were very malnourished. And once the war ended, they were able to finally eat and kind of get their strength back. That actually caused an epigenetic shift in their DNA, because the epigenome can be affected by environmental factors. Smoking, too much sun, breathing in really bad chemicals, like that can change your epigenetic makeup of cells in certain tissues. So being starved, being under, very malnourished, that will cause epigenetic changes in your body. They detected that in that generation. They were unable to detect a similar pattern passed down to their children, but they were able to determine a similar pattern after being brought down into their grandchildren. So it skipped a generation. I can't remember exactly all the details of that one. That was like 15 years ago.
Study, I'm not sure if it was epigenetics, but it's fascinating to me. It's also the basis of how they did it was great. So they took some sort of I don't remember what it was, and they gave it alcohol. And then they forced it basically out of alcohol, and then they stopped. And then they looked at some sort of marker, and the ones that didn't care about it again were fine, and the other ones would basically try to do anything to get it. So that study, I guess, helped figure out that like sometimes it is a genetic factor that makes people more prevalent.
An epigenetic or a genetic factor?
No.
Yeah, I don't know that study. I mean, it sounds like, looking at kind of addiction, right, and seeing if there's more susceptibility from organism to organism. Yeah. Yeah, I mean, you can look at, so that could be a genetic makeup. You could be getting maybe some allele from a parent that has, or several alleles from a parent that has a higher susceptibility for alcoholism, or dependence on alcohol. You could have epigenetic makeup of, different changes of the epigenome in certain people that could be affecting your tolerance and your dependability on alcohol, or any other kind of controlled substance, right? Yeah, I mean, I'm sure that if there's a study about it, I'm sure there's some really good papers on it. I'd be really curious to see that one. This is kind of complicated. I will go into how the NEB kit works and kind of more about the methylation. I think it's pretty important, because it's one of our more popular products, especially the capture panels. I haven't even gone into like custom panels or anything like that yet. So any questions on epigenetics in general? Kind of was very high level. Again, if you have any questions on this, ask your FAS. So some of there's a lot of, when you think of epigenetics, there's a lot of cancer, just because like the changes of the epigenome. The epigenome, if there are changes, if things are upregulated or downregulated, cancer can increase or decrease. So it's really dependent on the tissue type. It's dependent on the gene that's being activated or deactivated through the epigenetics, through the epigenome changes. So there's a whole, this is a huge field of study that is taking a lot of resources, because it is something that has really in the past like 20, 25 years has really like opened a lot of people's eyes on kind of how our genome works. So you can take that, yeah, take that in if you want. So what is NGS? We keep talking about NGS. What is it? So it's the current modern sequencing technology for rapid and simultaneous sequencing of millions to billions of DNA fragments, enabling high throughput, accurate, cost-effective genomic analysis. So you do get a base by base analysis of your genome or your RNA. So when you actually look at if someone has a mutation at position 1,500,000, blah, 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 you know, if there's you can actually see that single base resolution of a variant, right? It's high throughput. You can do thousands of samples at once. So this is like, even with whole genome sequencing, with some of our more powerful sequencers, you can still do like hundreds of samples of whole genome sequencing. And I'll go into why you can't do as many with whole genome, but just think that there's just so much data being generated. You're generating terabytes of data with doing this. When you do sequencing, all the technology that we have well, I'd say most of the technology that we have right now still utilizes cameras to take a video of DNA being sequenced. So that's how we actually can take biological material and turn it digital. And taking these big movies, these high resolution movies, takes a lot of data. So that's another thing to think about when talking to customers about NGS is their data capabilities and their data storage, right? It's cost effective because the higher the numbers, the samples, the lower the sample cost, right? As salespeople, you should know that, right? So really, there's really two different types of there's NGS sequencing and then there's Sanger sequencing, next gen sequencing and Sanger. Those are kind of like the two big main sequencing systems. Sanger sequencing is still used today. It's still a really good system. It's very, very accurate, right? It's like 99.99 something percent accurate, whereas NGS, you might get like 95 to 99 point something percent, but like it's extremely high accurate. People still use the Sanger sequencing for validations, especially when you're looking at gene edits. The biggest issue is that it's very low throughput. You can only do handfuls of samples, right? The way that this works, you have to make a primer, actually a primer set. You have to design your own primer set. The primer binds to the DNA and then it reads 1, 2, 3, 4 as it goes up, and it only goes up to about 700 bases per read. You get one read.
I did that. I was wearing
You're saying so you go bump, 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 bump. Takes forever. Can't do it. Yeah. So say you have a gene edit at position X, right? In chromosome, whatever. You build a primer, place it there, Sanger sequences, you get a validation, you see, oh, that A is now a T. I'm very certain that that's the case. Sanger sequencing will tell you that. So that's one sample, one reaction, one read. Took so long. Yeah. Took 20 years. Well, they're also using the gel system too, which was even more incredible. Yeah, you had these large gels. So they're like, yeah. There was, oh man, this technology is wild. All right. For all the people that don't know about the gel sequencing that Eric Lander and who's the other guy down there? Oh, I'm a bad scientist. I know the human genome guy, Eric Lander and the guy down in New York, the big boat. Blanking on his name. There was a sequencing war back in the day, so they actually had to do Craig Venter. Thank you. Couldn't think of his name. Yeah. Craig Venter wanted to take DNA and patent the genome, whereas Eric Lander was like, no, let's make it for everybody. But they also ran it on these big gels. They actually would do a PCR and amplification of a fragment up to a certain point and would terminate. and then they'd run it on a gel, and depending on the size, they would know that termination base. So for every single base that went up for billions of bases, they'd have to run a gel for each one of those bases. Now we do NGS. Quicker, faster, and cheaper. The first human genome, does anyone have a thought on how much it costs to sequence the first human genome? Yeah, it was upwards of $1 to $3 billion to sequence one human genome. How much do you think it costs now to sequence a whole human genome?
200 at list price. Cheaper.
Yeah. It's still expensive. It's still, you don't want to have like whole genome sequencing as like a diagnostic for a patient, because no one's going to pay $200. I mean, some people might, but yeah, exactly. So it's still expensive to run like a whole human genome sequencing per sample. That's why we have Twist. So this is really expensive at scale too. So that's a very poor system for like high throughput. Just some key terms for people to understand. So NGS library, it's a collection of DNA fragments prepared for NGS sequencing. So anytime you start talking about libraries, we talk about two different type of libraries at Twist. Does anyone know the other one? Yeah, so they have like variant libraries, right? That's a little bit different than what we do. We have next gen sequencing. Yeah. No. What's that? So yeah. So the variant libraries are basically just fragments of DNA that have synthetic or introduced variants. You throw that into a cell and look at it expressed differently. You do a screen that way. So that's one product that we offer, not in the NGS world. But in the NGS world, NGS libraries are fragments of DNA that can be sequenced. DNA RNA library preparation is the process of making those libraries. Sequencing read, this is what actually gets generated from the high throughput sequencer for in the terms of like short read sequencing, like Illumina or Element. You're usually talking about single read or paired end read sequencing. So 2x150, you'll see that one. No one really does single end sequencing anymore except for Ultima. Coverage just means how many times that nucleotide is read or sequenced. So that's all it means. You see 50x coverage on something, that's like on average how much that one base is read. That adenine is read 50 times, right? So usually when you look at low coverage or like low pass or I get low coverage whole genome sequencing, you're looking at like only like 1 to 10. Not very reliable. I wouldn't even talk about the 96-plex. What is this discontinued product that we have? Higher coverage for whole genome sequencing, you're looking at like 30 to 100x coverage. You generally don't see a lot of people doing more than 100x coverage for whole genome sequencing because it's just, again, it gets really expensive to sequence 3 billion bases. Variant calling. So this is good for identification of genetic variants. You might hear the word SNPs, single nucleotide polymorphisms. Indels, people have started writing this as one word now for some reason, but it means inserts or deletions, right? That just means like you get a chunk of DNA put in or you get a chunk of DNA taken out, and that's bad. You don't want that. You compare it to a reference genome. Multiplexing, you'll hear that quite a bit, which also usually is in tandem with indexing or barcoding. That's taking tagging different samples, not the molecules, but the samples with a unique DNA sequence that's attached to the library. And that allows for multiple samples to be sequenced at the same time. So that's when you start talking about pooling or multiplexing. Then that's when you start getting into our UDIs that have barcodes, FASTQs. So that's just the raw sequencing data that comes off the sequence. Actually, what comes off the sequencer is technically a base call file, BCL, that gets converted into a FASTQ. So there's a process called BCL to FASTQ. Base calls are what's physically read on the machine. So there's a camera looking down on a flow cell, and each time it flashes, it takes a picture and takes that data and turns it into what's called a base call. that there is an algorithm that takes those flashes of lights and turns it into digital data. And it's called a FASTQ file. So you'll hear about that a lot, FASTQs. Yeah. Well, in the context of whole genome sequencing, if you start multiplexing and combining a bunch of samples into one sequencing run, there's only so much sequencing capacity. There's only so much a sequencer can sequence. There's a limited sequencing capacity on a sequencer. So if you flood the sample or flood a flow cell with too many samples, you're not going to get as much coverage as you would like. You can still do that with whole genome sequencing. You can, if you're looking at kind of lower coverage, right? Depending on the sequencer. If it's like a NovaSeq, right? A NovaSeq will give you a lot of data. So then maybe you can multiplex, I don't know, what, 30 samples on a flow cell, maybe? 64 per flow cell. For 30x? Machine, what coverage you need.
How much rainfall I learned it was, you have an acre of land, and the acre is your flow cell. And an acre, the flow cells will have different amounts of land. This, if it has 30, if it has an acre, you could put, if you have one huge house on it, or you could put 50 smaller houses, and that could be like samples, but it depends how much land you need to make that one house rent. So if you do hold you do a microbial, there's all kinds of room, right? You could do all kinds of houses. You get quarter acre, you could have 1/8 acre, whatever. But you need to know the size of the flow cell before you can determine how many samples you can then put on the flow cell. How many samples can you multiply? You have to know the size of the flow cell and the max of it before you can determine it.
Okay. So in my head, I'm thinking, why wouldn't everybody who has more high food but just do multiplexing to save money?
Exactly.
Not everyone needs a NovaSeq, right? Like if you just have a couple samples, you don't want to spend all that money. Use a little sequencer with a little flow cell, and then you concentrate on, for example.
And so that's why I'll get to it later. But when we start talking about hybrid capture and targeted sequencing, you want more samples because I'll make it more your sequencing power. Your sequencing power is like you said, is fixed, right? There's only so many reads to go around, and that's always the case. So then if you pack more samples into that, you'll get more efficiency of your sequencing.
You're paying for every run.
Paying for every run.
Yes.
And how much is a Novus full S4 flow cell?
It's right now, $17. When they first came out, it was
Like 30. Yeah.
Too, so to hit go $60,000 for one.
Yeah.
So, back then it was 48
for whole genome sequencing.
For 2 flow cells, 24 per flow cell. And it was $30,000 per flow cell.
Now it's 17, which is pretty good.
Better. Actually, and that's on the X plus.
I was gonna say the X plus. Yeah. You can sleep through this because you know this already. But I'm just yeah. Yep. I just want to go through what sequencing actually is. Like Lumina has a great video of how the sequencing actually works on the flow cell. So I'm just gonna play this. I think this is super informative and it would be super helpful. Everybody hear that okay?
The Illumina sequencing workflow is composed of 4 basic steps: library preparation, cluster generation, sequencing, and data analysis. There are many ways to prepare libraries based on sample input type and sequencing application. All library preparation methods add adapters to the ends of the DNA fragments. Through library preparation, these adapters contain regions such as sequencing initiation site, indexes for molecular barcoding, and regions that will bind or hybridize to complementary sequences on the flow cell. Clustering is a process where each library fragment molecule is amplified to create a cluster of identical copies on the flow cell surface. Illumina utilizes 2 flow cell architectures: random flow cells with a lawn of oligos over the entire surface, and patterned flow cells with oligos only in nanowell. Newer Illumina systems utilize patterned flow cell technology to create clusters at greater density and efficiency. Flow cells are coated with a lawn of two types of oligos. Cluster generation occurs when library fragments hybridize to the surface of the flow cell. DNA fragments are bound to the flow cell substrate through hybridization of complementary sequences. After libraries are attached to the flow cell, the strands are now clonally amplified through a novel amplification chemistry called exclusion amplification, or XAMP. After the polymerase creates a copy of the hybridized template molecule, another copy is made by hybridizing to the second type of oligo on the surface, creating two identical copies tethered to the surface of the flow cell. The process is then repeated many times per cluster, resulting in clonal amplification of all the library fragments. XAMP performs this process.
This is just saying that your library gets put onto the flow cell and it just gets through, like similar to like a PCR, right? PCR amplification, similar to that. It gets amplified multiple times over and over and over again. So each one of these clusters, each one of these, you saw like the circle, that was the pattern flow cell. So in each one of those nanowells, there is the same exact sequence in each one of these micro wells or nanowells. Yeah. Yeah. Well, what's even better is some of these newer systems, actually all of them now, they do it on board. You used to have a separate system called the CBOT, and CBOT was, yeah, and so that would do all the amplification, then you have to take it off and put it on. It was a mess. So nowadays it does it all on board.
Repeated many times per cluster, resulting in clonal amplification of all the library fragments. XAMP performs this process quickly, efficiently, and in real time. After amplification, the reverse strands are removed, leaving only the forward strand. The 3' ends are blocked during the removal of the reverse strand.
Like this, and how often does that happen that like it's wrong and then so your results are wrong?
Yep. So there is something called amplification bias or sequencing bias. So there's two different steps where errors can be introduced into your process, right? There's either through PCR when you're doing the library preparation, so incorporation of incorrect bases through DNA amplification. There's also an amplification step here, right? So anytime you get an amplification, there's always the risk of incorporating the incorrect base. And though Illumina has done a great job at, optimizing their chemistry so you don't get any or get minimal integrations, but you do get errors that are introduced that can happen. So you might not necessarily get the same exact fragment throughout the entire lawn. You might get errors at like a one, I don't know how, it's like 1 in 3,000 Q score. Yeah. Yeah.
There's a very small amount of the little part that's then informatically to figure it out.
Yeah.
Most of the time.
Yeah. There's always error that's introduced. There's always incorrect bases that are introduced, but there are ways to kind of filter that out. And Twist has products to help improve that metric as well. So, hi Jimmy, can you hear me okay?
I can, yes. Thank you
Hey, welcome.
Yeah, thank you for having me, setting up Zoom.
No problem. I'm using this new microphone, so let me know if you can't hear me very well.
No, it sounds great.
Cool.
To prevent unwanted priming during sequencing. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, a mix of fluorescently tagged nucleotides are available for addition to the growing chain. Only a single fluorescent base is incorporated that is complementary to the sequence of the template.
A's to T's, G's to C's. Always.
The clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called Illumina sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. Illumina sequencing systems have moved from a 4-channel detection chemistry to a 2-channel detection chemistry. 2-channel detection chemistry improves efficiency and reduces sequencing runtime. New XLEAP SPS chemistry further improves the speed and accuracy of the 2-channel detection.
So this might be getting a little too technical, but essentially there are the old way of doing this was each one of these bases had a different color, and it would flash green, yellow, I don't remember all the colors, orange, but there's all the blue. There's all these different. There's 4 different colors that were illuminated once they incorporated and then flashed. So you would actually physically record. It'd be a lot. You'd see them like twinkling, like twinkling stars on this thing on a flow cell, and you just see them twinkling, right? And that'd be capturing the sequence. Now there's this 2-chemistry system or 2-color chemistry system. So you're looking at either red, green, yellow when they're together, when it's one, or nothing. So there could be issues that come up with the sequencing run when you see nothing, right? So it assumes nothing is a C or a G. G, yeah. And then yeah, it has its pros and cons, but
approach through an improved lock and linker and a new polymerase engineered to incorporate nucleotides faster and with higher fidelity. This delivers up to 2 times faster incorporation rates relative to standard
actually it doesn't work
SBF.
very well.
All identical sequence strands in a given cluster are read simultaneously. Hundreds of millions, up to billions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step of the example, the index 1 read primer is introduced and hybridized to the template. The index read is generated similar to the first read. After completion of the index read, the read product is washed off. The template now folds over and binds the second oligo on the flow cell. Polymerases extend from the second reverse strand oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the original forward strand is removed, leaving only the reverse strand. Index 2 is read in the same manner as Index 1, with the introduction of an Index 2 sequencing primer. The Index 2 product is then washed away. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. This entire process generates billions of reads representing all of the fragments. Sequences from pooled libraries are separated based on the unique indexes introduced during the library preparation. Based on the library preparation and chosen sequencing application, downstream processes can either assemble sequences to reconstruct the original sample without the need of a reference sequence, map and align sequences to a reference genome to identify regions that differ from the reference genome and detect variations, or count molecules via sequencing reads to assess abundance in gene expression.
RNA.
This omic data can be easily centralized within the Illumina connected software ecosystem,
Blah, blah, blah.
which integrates.
The rest of it just kind of goes into their analysis, like the onboard analysis system. Any questions about how that works? Yeah. We can make those libraries that get sequenced. Yeah. So our library preps are the upstream where it's talked about those fragments that are attached that look like they're called forked adapters or Y-shaped adapters or Illumina adapters, whatever you call them. We have those available, and we have the whole system that can make that library to be sequenced. I just think it's just kind of important to know just kind of the basics of how sequencing works, because especially the beginning where it talks about the adapter ligation and whatnot. So I don't really want to go into how PacBio sequencing works in terms of like the actual on the flow cell or the smart cell. It just know that it takes a long molecule. It does something very similar actually to Illumina. It's just instead of having a polymerase that binds and amplifies, the polymerase is bound to the smart cell, and then the fragment just goes around and around and around. And it has kind of a similar similar process in terms of like fluorescence. ONT is the one that I'm not going to go too much into ONT and how this works, but essentially it instead of flashing a light every single time it hits a base, it goes through this pore. It's a physical opening on this like really tiny molecular membrane. It funnels the DNA through and tests electrical charge as it's going through. And so it can test the as it's going through the pore, it'll say, hey, it has this kind of electrical profile, so it must be an A. And then, you know, there's issues with the ONT system, so it doesn't handle it's called monomers, or essentially what long chains of the same exact base. So it'll just kind of flood the system. It won't work very well. So those are just different sequencing technologies. The main ones that we see out there, the Illuminas, the PacBios, the ONTs. You'll also see elements. Elements out there have a very similar process to, they have a different amplification and kind of flow cell system, but it's all based on fluorescence as well. The nanoball technology, yeah, I got that here. This kind of looks like an octopus kind of thing, or flying spaghetti monster. You know, it has all these pieces hanging off, all these tendrils, and that gets sequenced. But you don't really need to know too much about that. Where does Twist fit in? I guess that's the $1 million question. Anywhere we're green right here is really where we fit in very well. So the customer does the nucleic acid extraction, which we all did today. Then you do a library preparation, either DNA or RNA. We either do DNA or RNA in various forms, right? We have like a low input or cell-free DNA kit. We have a methylation detection kit, which is also DNA, but also we have RNA library preparation kits. They can do an optional target enrichment. One thing that I keep seeing people talk about is like the full workflow. Technically, this is an optional step for people to do. It helps and it makes us a lot of money and it makes them have more efficiency, but it is completely optional. You don't have to do a target enrichment to do NGS. So the full workflow, if you say to a customer, the full workflow, they'll think all of this, but really we have the full library preparation workflow with an optional target enrichment. But we really want to push target enrichment because that's where we make money. So again, I'm sharing all this with you guys. So where I marked with the green is products labeled or products supported by Twist. Any questions there? We already did this, right? So this is just how people do nucleic acid extractions on a more clean and a more professional way. So you have different fluids, you have different sources, you have cell-free and circulating tumor DNA. That's a big one. So that comes from blood or plasma, right? Cells are really big. Tissue type tissues, what we did today, that we consider a tissue. Tissues can come in essentially two different forms, either freshly frozen, which means that they just take a chunk of tissue and freeze it, right? Put it into liquid nitrogen. They could take cells right or tissue right from the source, which is called the primary or biopsy. Or you could do FFPE samples. You might hear about this a lot. Formalin fixation. Does anyone know what that means? Anyone know what FFPE means?
Stands for
Oh,
Or what is FFPE? Like what is it?
just fixing it so it locks in the profile of the DNA or RNA into, usually they put it in a paraffin block, so then it lasts. Like you go in these labs and there's blocks for like 20 years, right? Just sitting around, and then they just use very like micron slices, and then they can actually then extract it.
Yeah.
That's good DNA. So having a really good library.
Yeah. FFPE is used quite a bit for archival or storage of samples. What it does is it basically freezes the cells or tissues in time. It stops all cellular processes. It's the same stuff or very similar stuff that they use to embalm people when they die. And basically it's formalin or formamide, very similar process, very similar stuff. They put it in your body because it stops or slows down the decomposition of your body. It stops or slows down cellular processes. So it's a way to take a snapshot at that time of the cell process. Yeah, I mean, you can't Here, you're looking at small pieces of tissue, so the FF, the formalin or the formamide can penetrate deeper, right? And that's why you can do it with small pieces of tissue. With humans, like there's not much, they just put it into your bloodstream and you don't have a lot of surface area there, right? So then it slows down the process.
Trying to like edge it, like cancer.
Biopsies, yeah.
Live.
Yeah.
Do they bother? Do they still have to like flash freeze in the night?
No.
again, or
No. Depending on what's happening, if it's going right to the lab, they'll just put it in. Likely what they'll end up doing is literally just put it in a lysis buffer and ship it over real quick. If they're doing DNA work, especially because DNA can just be stable. DNA is very, very stable, even in FFPE, right? I mean, people sequence DNA that's like thousands of years. It's damaged. It gets damaged. Yeah, but it's still sequenceable. RNA is a little bit different. If they take it out of the body, they have to put it into a preservative like Trizol, or there's other preservatives that they have out there. Yeah, because RNA is very fragile. Single-stranded RNA will break down, and so whenever you hear people talk about RNA manipulating it, there's a lot of potential for degradation of RNA. Okay, so then you take how much time? It's 11. Okay. So you take all your sample types. We got to do what we just did. We got to break open the cells. You have this lysis buffer and cells and tissue floating around. You load this lysate. What we did, this is similar to the paper towel. You have to collect the DNA. It's a little bit different because the DNA actually binds to the paper towel or silica membrane in this case. And then you use some water to elute off of it, and then you have isolated DNA. So it's very, very, very pure DNA doing it this way. We don't currently offer a solution. I guess we're working on one at some point for our apps lab. So any questions there? I know I kind of breezed through that real quick.
Sort of favor certain purification kits over others, or pretty much
we're fairly agnostic. There's certain ones that I don't like. Haven't had really good success with like the NEB Monarch kits. I really like the kind of the Invitrogen or the Thermo ones that are pretty good. Kaigen ones are pretty good. It all depends on also what you're using. There's different extraction types for different cell or sample types. There's always a different, like there could be like a total nucleic acid for like all sample types, but those tend to be kind of funky. So usually there's extraction methods for different inputs. So. So library preparation. I know we kind of went into this a little bit with the video, and I've been hinting at the process, but this is actually the step-by-step. This is important to kind of know because we sell this. This is something that we sell. We sell multiple flavors of this. So you have to break the DNA into smaller sequenceable fragments. Yeah. Oh, sorry. A little better. Okay. So there's two main modes, kind of mainstream modes of breaking DNA up into smaller fragments. You have the Illumina sequencing, unlike PacBio and ONT sequencing, can only sequence fragments that are up to maybe 500 to 600 bases. They're pushing it at 650. But so you need to digest it into smaller fragments. People generally sequence fragment sizes about 350 bases long. Very small. Compared to 3 billion bases, you're bringing things down to 300, right? So it's pretty incredible what we're doing here. You have to fragment it up into chunks using either mechanical force. You can use grinding, some people use back in the day, but most people end up using something called sonication, which uses a high energy sound wave to break up DNA. It's pretty cool. Very low throughput, very expensive. Most people don't want to do that. So they want to move over to an enzymatic method, which is what we provide. We have a mechanical fragmentation kit, but that just literally just means that we skip the fragmentation altogether and let the customer do their own fragmentation. So that we have an enzyme that will come in and non-biasedly or unbiasedly chop up the DNA. We always have customers ask about our enzyme and how we fragment. When I do a comparison to mechanical fragmentation, a lot of people still think enzymes will biasly chop up DNA, and yeah, there is a little bit, which means that the enzyme will cut at a certain sequence, right? A very specific certain sequence. What's nice about our system is the bias is just about as low as mechanical fragmentation or sonication, which is completely random, 100% random. That comes up a lot. So if you have questions about that, bring it up to your FAS.
Is there a big difference between Twist's enzymatic fragmentation and like Illumina's Nextera?
Oh yeah. Oh yeah. So the Nextera system has a different type of fragmentation, right? The Illumina system, the Nextera system uses something called tagmentation, which recognizes certain sequences and introduces the adapters into the library. And that is, if you look at a trace, if you look at the actual genomic or the library profile, it's not great. So this has a lot less bias because you're just chopping it up versus integrating things into it.
Same way, just a different way to do it.
Yep. Yep, exactly.
Just be to say that ours is about the same as the mechanical.
I have a nice figure that has this is getting really technical, right? So we look at chimeras. Chimeras are fragments of DNA that have similar ends that could potentially self-ligate from different parts of the genome. You don't want that because then you can't merely make an association of this part of the genome to that part of the genome when they come together. So how those happen are having similar cut sites, similar sequences, similar biased cut sites. And some enzymes, some mechanical fragment or enzymatic enzymes will have that bias. We compared the chimeras of our EF 2.0 kit to a mechanical fragmentation kit, and they're roughly the same. You get a little bit of overhang. Yeah, I don't believe it's blunt ended. What's that? I'm not sure exactly. Yeah, I'm not sure. I don't believe it's blunt ended. Yeah. We do an A-tailing, yeah. But I thought the enzyme would leave an overhang, no? Yeah, right. Right, and that's why sonication works so well.
Like Nextera or other enzymatic frags?
Yeah. It can be. It's wasted data. Yeah, you have to throw it away. And there's ways to like detect it through like bioinformatics, but then it just gets tossed. NEB has a particularly higher chimeric reads, bias for some reason. I'm not sure why. All right. So you obtain relatively uniform sizes. That just means you chop everything up to around the same size, 300 bases, 350, 400, whatever the customer really wants to do. You chop it up to that kind of size, this insert here. Then you have to do something called an end repair and an A-tailing. End repair just takes DNA that has an overhang. So basically, if DNA doesn't completely overlap, if it's kind of like, if there are overhangs, basically double-stranded DNA with an overhang one way or another way. So is it? Yeah, that's what I figured. Yeah. So I mean, it's MNase based. So then MNase is non-discriminate. It'll just chop, and it could leave some overhangs. So then the end repair will come in and fill those ends. I don't have a board, but essentially, instead of having a blunt end means the DNA is completely overlapped, whereas an overhang means that it's slightly shifted over, so it doesn't have full complementary double-stranded. Well, that's what the end repair does. End repair does and fills those gaps in. And then you have to add an adenine to the end of your library. So then you do have a single base overhang. The adapter that we have, what's called our either a full-length adapter or what we use more often are these universal or stubby adapters or partial Illumina adapters, all the same thing. These adapters have a T overhang. that T anneals. I know, I'm sorry, this is kind of small, but this T will anneal to this A. And that's what we call TA ligation. And that, you'll hear that quite a bit. So there's really two different main kind of library preps that are out there. There's Nextera, which is not a TA, it's a tagmentation-based system. And then there's the Twist, or another name for it is TrueSeq style, which is also an Illumina term, or a TA ligation-based system. That's what we offer. We don't do any tagmentation at Twist. We do a TA ligation-based adapter ligation. So the T overlaps with the A, anneals, then you have a partial Illumina primer or library. I'll go into UDIs a little bit, probably either later today or tomorrow. The UDI, it's really kind of, it's a little bit more complicated, and I really want to kind of drive that home. So I'll have like a whole kind of thing on that. You do a PCR amplification using primers that have the barcode on them. The barcode then, the primer then amplifies the full fragment with the barcode. And that's how you can get 384 different samples, or 384 unique samples, or up to 3,072 unique samples, individually barcoded and ready to be sequenced. Any questions on that? This is the tenet to library preparation. This is kind of the very basics of library preparation, but all of our kits do this in some form. So it's probably pretty good to understand this a little bit. So if there's any questions, please let me know. Okay. All right, finally getting into hybrid capture. How does hybrid capture work, right? This is Twist's money maker, right? This is our big money, because we own this product fully. We have full rights to this product. This is our own product. We have large margins on this product, so we can play around with pricing. We can play around with like how much money we can make based on this. This is really, we want to drive. We do have library prep, and we can do whole genome sequencing just fine, but we really want to press hybrid capture. All of them. Maybe. Yeah. We have two hide methods. Mm-hmm. Yeah. So the library preps, it's somewhat confidential, somewhat not, but the library preparations are OEM'd, which means that they're all from third parties, except for our cfDNA library prep kit. We don't have as much margins on those because we buy them and then resell them. But our cfDNA library prep and our hybrid capture reagents and our probes are, none of that is, I don't want to say none of that is OEM, because there might be some products that are OEM, like the primers. But for the most part, we own that whole process, so we can really bring home some money with that. How it works is you have the library, right? You have the full library, which has those P5, P7 primers, those regions that were mentioned in the video. It has the barcodes that were mentioned in the video. It has the inserts. So this insert here is that 450 or so bases that want to be sequenced. You have barcodes that you can pool samples together, so you can uniquely identify each sample. You take a complementary pool of DNA fragments. These are short fragments. They're 120 bases long. They're double-stranded. That's very important. And they're biotinylated, which means that there is a molecule called biotin. I think a lot of people use that in kind of eye products as well, right? Biotin, does it like puff up your eyes a little bit more, or like skincare? Yeah, I don't know. Do people use biotin for like skincare? What's that? For hair. Okay, for hair. Yeah. So because it's sticky, right? It helps make your hair nice and glossy or something. I don't know. So yeah, I mean, it's a product that's found everywhere, right? So you can use that, you can put that on DNA. This biotin, the sticky molecule, will stick to something called streptavidin. Streptavidin and biotin form the strongest binding force known to man, known to humans. Covalent, yeah. So it's very strong. It's a very strong bond, right? So once the biotin and streptavidin meet, you cannot separate that. There are chemicals that can potentially do that, but we don't want to do that because that'll destroy your DNA. Both of them. They're double-stranded. So since they're double-stranded, you can. Yeah. So since they're double-stranded, there's a biotin on each 5' end. They get separate. They get denatured. So now you have two probes, one for the sense and one for the antisense. Yep. So that's one thing you can talk to customers about since they are double-stranded and both strands are biotinylated. They can capture both fragment or both antisense and sense strands. So you get double the capture efficiency. Yeah, so basically what you're taking is you're taking a whole bunch of library, a whole genome of library, all 3 billion bases of library, and then you're doing a capture. You're removing targets that you don't want. These biotin labeled oligos are defined by the customer. They bind to certain targets in the genome, genes, genomic regions, RNA regions. They will physically bind to the DNA, isolate just the region that you're looking for, remove everything, and then you're down to what's called an enriched library. So that's the tenet of hybrid capture. Uses probes to isolate fragments of targets that customers are specifically looking to interrogate. Yep. Depends on the size of the panel. So the error is something that's called off target. You might hear that quite a bit. Our best performing hybrid capture panel, you get an off target of roughly 10 to 15% of the genomic or the sequencing reads will go toward off target binding. So that just means these probes will be binding somewhere else in the genome unintendedly. It happens. You can't. 10 to 15% is our best performing panels. You just remove them off. Yep. So that's like our exome. It's about 10 to 15% off target.
Is that industry standard?
It's better than industry standard. Oh yeah. Yeah. Usually it's like 20%. Yeah. So what that means is you have more of your targets coming through, so you don't have to sequence as much. Any questions on this? This is another big tenet. So the library prep and hybrid capture, those are our big bigs. Process it, think about it if you have any questions. No, but it is something that we are coming out with. It's basically just EF 2.0, but with our own products. I mean, it has a lot of similarities in terms of the workflows. It just has our products introduced into it. It's still a TA ligation-based system. Has our own ligase, has our own polymerase, has our own ERA and repair A-tailing cocktail. Fragmentase.
So I was something the committee about they want to make it worth the while, so whether it's cost, but also they want to. And one of the things over and over again. Break everything down the same side of things. Software instead of just looking pretty way.
Oh yeah. If you only have to have one condition for all like DNA size types. Above your pay grade. We have high AT stuff. Does everyone know Paul Fair? Okay. What's your title? Whatever that means. What is it that you do here? What would you say you do here? Yeah. Yeah. Um, Paul's great. He's also really good with automation stuff, but don't talk to him about that because he's not in that role anymore. Yeah. Yeah. And bravos. Do bravos. Oh geez. Yeah. 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 They have an interesting system.
What you're referring to is different. So, yeah, anyway, now they reinvented it. Oh, you actually run some tickets.
Oh yeah, I remember Broad had one of those. The Broad had one. Yeah. Yep.
So we got to move all the places where we had all the things.
I'm going to go through probably two more slides and we can take a quick break. So we're kind of the time is a little bit funky right now. So okay, so we kind of know about this. We just watched a video, but this is more of a takeaway to know that once you're done with the enriched library or whole genome library, you take that and put it onto a flow cell. The barcodes, the adapters that are at the end of these fragments, so these colorful pieces here, specifically the orange and green, those are the sequence, or that is the adapter that is very specific to the system that is being used, right? So you can have adapters for Illumina sequencing. Those are what they're called P5, P7 adapters. You can have adapters that go on to a PacBio. Those are called SmartBell adapters, SMRT. You can have adapters that go on to an Oxford nanopore. Those are just, I forget what they call theirs, but there's just ONT adapters. And you can have adapters that go on to the Avidity element system. So every system has their own adapter system that goes onto that sequencing machine. One thing to note is we really only sell, for the most part, we only sell ones for Illumina sequencing. A lot of other systems out there, a lot of other next generation, fourth generation sequencers like Avidis or MGIs or Ultimas, they can take an Illumina sequence library, Illumina library, and convert that into a library to use on their system. So they just add basically another adapter to that. There is one, does anyone know what's the one caveat or one exception to that? What Twist does sell? It's not for you. We have an element library prep kit. Adapters. Yeah. We worked with them pretty closely. So we have like an element library prep with element adapters. So that would these colorful ends would be for element. That's like the only one though, as far as I know. Right, Paul? We don't have any other ones, any other library prep kits for other sequencers.
We have to see.
Oh yeah, I guess.
So there's PMC, which is PCR. And recommend everybody buy our kits. They have really good ligates that and they put their special adapters on there. And they're like adapters, and they can combine the top strand with the bottom strand, and they get really good fidelity of being able to make these calls, even though they're so if you're using that's the card, it's not our adapters. So I have the, it really could be all. Yeah, we work with that also because as you mentioned, like there's we don't, but it's like
Ion Torrent.
maybe
I don't want to work with Ion Torrent. Don't make me work with Ion Torrent. I'll curse you if you make me work with an Ion Torrent library. Please no. Yeah. I saw, you know what? I saw the Ion Torrent yacht. Yeah. Then in that sequencing war in 2012. Those are fun days. Those are fun days. I was in Miami and I saw the moored. I was just saying I saw the moored up. They had a yacht. I forget the guy's name that owns it, but it was moored up in Miami one. I was like, oh, that's cool. But yeah. Yeah. I think so. Yeah.
I guess.
Yeah, this time to get something better, I guess. Analysis. So, okay. The data analysis gets really complicated really quickly. So I don't want you to take everything here. Don't worry about too much in terms of like knowing everything here about one thing you should know though is that like there's primary, secondary, and tertiary analysis. For this training, really we end up stopping at like secondary analysis. So primary analysis is really just looking at raw sequences and kind of how it performed on the sequencer. You're talking about something called a Q30 or Q40. You might hear something Q50. Those are just quality scores that measure the error. As Vivian was mentioning, there is always an error rate associated with DNA sequencing. Wrong bases can be incorporated into the sequence. And when you're looking at Q30s, which is what Illumina measures their quality reads by, that just means it's such a stupid number that they do that. I hate this. It means it's 1 in 1,000 bases have been incorporated incorrectly, or 99.9% base call accuracy. They should just call it that. I hate the Q system. It's stupid because it doesn't really make sense to me. Q40 means that there's 1 in 10,000 bases that are incorporated incorrectly. You're starting to get sequencers like the PBM-seq, parts per million, parts per billion. So you're starting to look at like Q50s that Element claims they can get, which means they can get 1 in 100,000 bases are incorporated incorrectly. And then you could even potentially get like up to Q60s for some of these things that are 1 in a million bases incorporated incorrectly, or 99.9999% accurate. So you look at this, you're like, it's not just 100% accurate, but when you're looking at billions of bases, 1 out of a million is still pretty high. So, and especially when you're looking at a billion bases, 1 in 1,000 is certainly too high. So that's where Illumina starts.
Take care of it. You go deep enough, you're going to see if it's right or not.
The more you sequence, the more error gets introduced.
If you oversequence it.
If you oversequence, yeah. Well, no matter. So there's ways to handle this, right? And I'll talk about our adapters, but UMI, so basically tagging the original molecule to make sure that that is the original molecule or not. So, so there's different metrics to look at for oversequencing. The one thing I look at is if so there's a measurement called duplication rate. So at some point you're sequencing everything, and at some point you're going to start sequencing the same exact molecule more and more and more and more, right? So if you have the same exact molecule sequence more and more and more, that's zero data. You know, if you have the same molecule over sequenced, you're just going to throw away that data because you already have the information you need. You don't need any of this extra sequencing on those molecules, right? So then you throw that data away and you're left with, you know, over sequenced expensive data, right? So if you look at duplication rate, so duplicates, so it just means what percentage of the reads are the same exact molecule. If you're over like 5%, then you're not looking too good, depending on what you're doing though. So if you're looking at, there are certain applications where you want a high duplication rate. It's like 30 to 50% duplication. So then you're sequencing at like 100,000x, million x. Generally for hybrid capture though, you're probably going to be targeting around 1,000x, más o menos. You could if you're talking about like low allele detection or high, they need high sensitivity if they're looking for really low detection of alleles, allele variants. So that's like liquid biopsy, MRD, low detection, right? These people are looking for one in a billion. They're looking for a needle in a haystack. That variant might be floating around in a body somewhere, and they're trying to detect it. So they need a sequence super, super high to make sure they hit that. But if they do that, the duplication rate goes up quite a bit. You can use a UMI system, and I'll go into that a little bit later, to tag the original molecule. So then you can say that original molecule that detected that variant is not introduced through either PCR or sequencing errors, because it's that original molecule. So I'll go into that more later, though.
Sure, I got that. You generally speaking, rule of thumb, 1,000x times the size of the panel would be what you would expect would be a good coverage of that panel.
It depends. I would say that's it's a good place to start. It depends on the size. I would say for exomes, right? That's pretty high. I would say that's a little bit too high. For exomes, you're over sequencing.
Always ask.
Right?
So
yeah, I mean, for exomes, you're looking at like minimum 50x coverage, 30 to 50.
Larger 50x.
Right. Yeah. If you're looking at some
it's a hard question, right?
Yeah.
You want to give them a starting, like the starting point is, you always give a disclaimer. Like, you gotta figure that out, but we would say start here.
Yeah, I always say like over sequence your pilot or your first one, and because you can always downsample, and then once you downsample, then you can figure out where you want to be. So like whatever, like if you think you need to be here, like I wonder if I need like 200 or 5, do what Patrick said, like it's 1,000. Downsample with different coverages and then
yeah, and it really, yeah, if you're looking at, so here's a couple applications. So there's two types of cancer detection kind of limits or studies in general, right? So there's germline detection. Germline just means every cell in your body has that exact same mutation because it comes from a germline, comes from your parents. Or there's somatic detection, which means it could be a small grouping of cells in a tumor. So you need to sequence more because you're also sequencing all the other stuff in that tumor, right? You could have that 1 or 2 or 10 or 1,000 cells amongst many normal cells within a tumor. Because tumor cells could be, you take a biopsy, you could be scooping out some regular muscle cells or that tumor, right? So there's a mixed population. So you need to sequence even more to make sure you hit that detection limit. In germline cells, you don't need that much. Germline, you need like 30x maybe, because you're basically looking at either 100% one, 100% other, or 50-50, because parents of how genetics works.
That's what those like children's hospitals that do whole genome sequencing 30x.
It's fine.
Just because you're going to catch most of something that's hereditary versus cancer, like 100x.
At least 100x. Yeah, I mean, even if your frequency is like 5%, 100x might even be too small. because you're only looking for five variants there, right? All right, so there's a bunch of analysis stuff here that we can go through. At some point today, I believe, let me double check that. Let's see, just gonna double check something. I didn't put it in the schedule yet. 11, 12, 1. So at one our time, I asked Rebecca to come on, and she's going to talk about some of the analysis and some of the design stuff. And we have Jimmy on board. If you want to say anything about this slide, Jimmy, you know, if you can chime in here. But this is really just saying here that, typically you go from base call file, it comes off the sequencer, FASTQ files that are actual digital files that you can measure. You basically take FASTQs, those are the sequencing files, those are the actual reads, align it. So there's BAM, binary alignment. I forget what the M is. So basically you're aligning to a genome, you're taking your reads, aligning it to a genome, looking at variants off of that reference genome, or looking at transcription. So there's a different analysis downstream that you can do.